And welcome to PBA at Noon. I'm Paul Benwell, and I'm joined by my colleague, Sophie Caesar. It is our pleasure to be hosting this PBA at Noon in association with Kin Communications. Today, we are very excited to be presenting Sona Nanotech's CEO, David Regan. I think it's safe to say that Sona Nanotech, which trades on the CSE under the symbol SONA, has been one of the best performing stocks during this pandemic, and with good reason. They have developed a rapid point-of-care COVID-19 antigen test. The test offers a unique advantage over other lateral flow tests as it specifically detects the presence of the COVID-19 virus. The opportunity here is monumental in more ways than one. David, welcome, and thank you for taking the time to speak with us today. Feel free to begin your presentation, and once you're done, Sophie and I will be asking questions from the audience. Thank you.
Thank you, Paul and Sophie, and thank you everyone for joining today. I'm going to just share my screen and jump right into a presentation here. Here we go. That is on. And let's go to full screen. There we go. That look okay?
Yep, that's
Looks
good.
Great.
Great stuff. Okay, so we're Sona Nanotech, and fundamentally we make rapid biological diagnostics better. I'm going to walk through today exactly how it is we do that, talk about our current test as well as our other underlying technology, I should say, and other possible tests in the future. But first, I'd point out our safe harbor forward-looking statements disclosure that will be on our presentation. Jumping right in, a couple of days ago, I saw this headline, and it really struck me that testing centers are absolutely buckling. When you see lineups like this, fundamentally we're just in a very, very difficult time. Question is, why are hundreds of thousands of people lining up and waiting for hours to get tested and then waiting for days? These people believe they have symptoms of COVID. They think they have the symptoms. They've got, whether it's a cough, a temperature, a sniffle. That's why they're there. The problem is, of course, by the time symptoms are exhibited, they've probably already been contagious for perhaps 5 to 7 days. And in this slide, you see a stage of infection schematic that points out that there's a 5 to 7 day delay, we believe, between infection and when symptoms show. Now that's when folks are going to get tested, and that's a fundamental problem. However, using a rapid point of care antigen test before the subject has symptoms is how we're going to transform the current context for society and the economy. So let's talk a little bit about Sona and the history here. At our core, we make the key materials of rapid lateral flow tests. This has not, however, been a short road. It was about 8 years ago that one of our founders started down the path. And of course, necessity is the mother of invention. And we're seeing that in these COVID times in particular. But 8 years ago, this particular founder of ours was working in a lab, needed to carve out his own space away from a mentor in order to secure funding. And was told, well, you can't work on the same thing that your mentor is working on. So he went after a particular conventional wisdom within the lateral flow community, which was that gold nanorods are superior in detection, but you can't use them because they're toxic. By actually making them elongated, making them into rods, it makes them toxic as part of that process. And he wanted to challenge that conventional wisdom. And I think it says a lot about the ingenuity and interest of bright young minds wanting to challenge conventional wisdom, and we have him to thank for that, and the world hopefully to benefit. Looking at a quick timeline here on page 5, I've got it starting in January, but the reality is it started 8 years ago with a lot of research, and it culminated last October when things gelled together on the science side and the gold nanorods were working well. And we thought, okay, this is great. Now we had a period of time of assessment saying what markets should we go after here, because there are all sorts of tests we can do. Lo and behold, in January, of course, COVID hit, and the team recognized that they had not just an opportunity, but a mission here, a need to create a test that would rapidly, at point of care, detect COVID. So January was proof of concept, moved into feasibility, development, optimization. During that period, we were very, very fortunate to receive federal government funding through Antigen, the next generation supercluster, to help us accelerate the development of this timeline for this test. And this, of course, moved into validation, where we are now, and into manufacturing, which was already started in terms of pilot batches, et cetera. Key takeaway from this slide, of course, is that typically this would be a 24-month cycle, and we've done it so far. The team has done it, led by Darren Rolls, our chief scientist and president, and the team in the lab. That's been nothing short of extraordinary. So today, 4 things I want to talk about. First, our COVID test. Second, platform technology. Third, other tests. And 4, longer term, where we see ourselves going and how we can make a difference, we believe, quite tangibly. So our COVID test, starting there, we started with SARS-based materials to develop an antigen from the protein within the virus to generate antibodies. So this is an antigen test because it's developed from that antigen. But we leverage also antibodies in the biologics, not to get too far into it. These antibodies, of course, want to glom on to the spikes of the virus. That's their only job, is to go and attack them and connect with them. So we conjugate our gold nanorods onto those antibodies so that we can trace them and track them, find them. They're effectively little flags, and that's what makes the test work. And I'll get into more detail on that a little bit later. But that's at the fundamental level, that's how it works. Now, the benefits of this test are really fourfold. They provide early detection. Again, it's those first few days, the first week after infection, before symptoms show, when it can be deployed most effectively. And you can imagine whether it's a testing and tracing program that employed auditing of a asymptomatic population to find out before people had symptoms that they have COVID and are contagious. Doing that can go enormous length towards compressing the spread. Secondly, it's quick detection. Results are within 15 minutes. It's a very simple test, like a pregnancy test. Therefore, on-site results, it can be done just about anywhere. It can be done anywhere. It doesn't need a highly trained healthcare professional. It needs someone, a technician who's been trained in it, but it doesn't need to be a doctor or necessarily a nurse. And finally, it's a lower cost. And the quick story, I know I've only got about 20 minutes here, but had a producer friend who called up. He's shooting a television show, and he's got actors and crew who've come in from across province boundaries, and he's being required to do testing of all the contingent of the team at a cost of $225 per person. The problem with that is, that's one thing. That's not a lot of money given the budget, but he's then got to wait 2 to 3 days to find out whether they're clear or not. For him, the value of having instant results with a fairly good indication would be extremely powerful, and he'd happily pay the same price for faster results, arguably perhaps more. Looking at the different testing methodologies, because that's something that we can get confused by, all of us, given the media reports, and there's been a lack of depth in some of the mainstream media as to distinguishing between the molecular PCR tests, which are really the gold standard, which are done in labs, the serological antibody tests, which play an important role, but fundamentally detect the antibodies developed by the blood, such as IgG and IgM, to infections generally. However, it takes about 5 or 6, 7 days for the body to generate sufficient antibodies to be detected. Antigen tests, of course, as we've discussed, can detect a virus within a day to 2 days, we believe, and certainly can be done anywhere, and you get results quickly. Moving along, talk a little bit about our fundamental technology. What we have here is, a lot of talk around our gold nanorods. It's 3 things really. It's first and foremost the know-how of the team. Secondly, it's the right biological materials we've used in terms of the right antigens and the right antibodies that have been developed. And finally, it's our proprietary gold nanorods. You see that in the upper left-hand corner. We use lateral flow assays. Our devices can be multiplexed, meaning they can have multiple strips, meaning they can test. One test can be used for multiple biologics being tested for. And of course, it's easy to read, easy to interpret those results. Now, look at each of these in turn quickly. Lateral flow tests are like a pregnancy test. They're low cost, simple to administer, and easy to use. And these tests rely upon the capillary action of the moisture that's put into them to be pulled down across each of those test and control strips, which will then light up and indicate that A, the test has worked, that it's actually run through, and B, whether there's a positive or not. I want to move along quickly. So the secret sauce, of course, are our gold nanorods. At a high level, these are effectively markers or flags that enable a visual readout on our lateral flow tests because they're attached to the biological analytes, such as the antibodies or antigens, that then want to track and attach to the test line on that test. If they attach to that test line, it lights up. Now, these gold nanorods are better than the typically used gold nanospheres, we believe. One understanding is that because there's more of a sweeping action with an elongated device, you pick up more, you're more likely to pick up materials with it. In the case of a hepatitis B test we ran last year, an evaluation of our gold nanorods versus gold nanospheres, the gold nanorods showed a detection level that was a 2 times improvement on it. On the 4, you can see here on the right-hand side. So 100% improvement, effectively, going from it's 2 of the metric here, the UPEI per milliliter. But how is it that they're better? One of the key reasons they're better is that they're non-toxic. And the non-toxicity is compelling. It's not a nice to have environmental aspect. The fact is that these gold nanorods, when generated, they start as spheres and they're elongated as part of the process, and they become toxic as part of that process, killing any of the biologics that might be involved, whether it's on a test strip or whatnot. These gold nanorods have been also optimized to work well with our biologics and with the membranes you see in those lateral flow tests so that they flow through that capillary action down the test well. That's a fundamental requirement. It's not good enough to have these materials just conjugate well with an antibody and then attract and bind with the virus. They've got to actually flow down the membrane too, so that we can register it across the line. So that's one test we have, of course, the COVID test. In addition, we'll be constantly improving this COVID test and generating a version 2.0. But in addition to that, of course, there is a myriad of different areas that these tests can be used for: medical devices, consumer health, veterinary diagnostics, food industry, military, and first responders. Those are the various areas that we were evaluating before COVID hit. And once we've hopefully made a big dent in COVID, we'll be able to return to working on those further tests. But in the longer term, this is when things get really interesting, because over on the left-hand side here, you see these in vitro, outside of the body applications. We're only right now doing one application in this one area. And yet, many people would have perhaps come to the realization already that when we say that these markers, these gold nanorods, are non-toxic, they'd recognize they can be used for in vivo applications also. And that's when people, medical professionals, get really quite excited about it, when the light bulb goes on effectively, because that means they can be used for targeted drug delivery, photothermal therapy, whereby they follow an ingested biologic that then goes to a tumor and can be lit up by infrared from outside the body in order to do targeted therapy. This is all, and of course the cell imaging as well. These are all forms of targeted precision-based therapies. This is not the near term. This is longer term, where we want to go with this technology and where it's going to enable us. So very exciting stuff. This is the team, very, very lucky to have some great folks in the lab and on our scientific advisory team, guiding us from, in many cases, afar, but these are leading thinkers worldwide in lateral flow tests and in the conjugation of nanoparticles. Finally, we know that there's increasing evidence that there isn't going to be immunity to COVID, and yet we want to return to normal. This test, antigen tests in general, can play an enormous part in returning us to a state of normal, and that's very much our mission. So I'd now be very happy to take any questions anyone might have.
Great. Thank you so much, David, for the presentation. Let's go over some questions. We've had quite a few. One is congratulating you on your tremendous efforts to date. This is a game changer in response to COVID-19. Can you discuss your plans to secure FDA approval? And also, are you making a submission to Health Canada and other approval bodies around the world?
Yes, that's certainly a requirement of many buyers, that there be FDA and certainly in Canada, Health Canada approval of these devices. However, these tests are in vitro tests, so they're not going inside the body. So, lengthy trials are not required and whatnot. And in fact, the FDA in particular has developed an emergency use authorization protocol whereby they're accelerating timeframes. So we've certainly already pre-submitted to the FDA and have had a dialogue with them on what they'll need to see in our final submission. And the same goes for Health Canada. The question we get asked a lot is, how long is that going to be? We don't know. That said, we've got a strong dialogue with them. We know they understand the power of rapid point-of-care antigen tests. And they've put in place a specific emergency use authorization. So we expect a fairly rapid turnaround time on our submission when it goes in.
Well, that's excellent to hear. The next question is, if you could discuss the LOIs and factoring them in, can you give us a ballpark number of tests you hope to sell upon FDA approval?
Yeah, good question. Yeah, we've had a lot of inbound interest, as you can imagine, just from some of the journal references to our tests in development prototype. And so we've been taking stock of that, and many folks have asked to sign LOIs in order to register themselves as an interested party and effectively get in the queue. And that's continued to grow. But there are many of the largest buyers who fundamentally require an FDA or a Health Canada approval in order for their governance-wise just to process and proceed. So that's a key gating item for those orders. No matter how many tens of millions they're also contingent on as soon as you have that, we're ready to go. This is queued up. We haven't indicated the full value there, but fundamentally, what's important is that on day one, when approval comes in, when it's effectively on the market, that's when it counts, and that's when we'll start talking about that. I think the second part of that was how much we'd be able to sell at that point, what the demand would look like. I think our view is our view being everyone on this call has probably a shared sense of the fact that if our test performs where it has performed and where we think it will perform, the demand is going to far outstretch the supply in the near term. We have manufacturing that is ramping up, and we'll be able to meet that demand, but it's going to be easily, we believe, into the tens of millions, into hundreds of millions potentially.
So I guess that brings us to our next question. And obviously there will be, a ramp up going forward, but how many tests per week would you be capable of producing, let's say at this time, and then to ramp up to, those millions of tests?
Right. So, we have a technology transfer going on with one manufacturing partner now and working with several others. One's in Europe, several working with are in the US. They're frankly, many of them are in the same situation as the buyers who are saying, well, yes, you want capacity in our plants, but we need to see that you've actually got something to sell. So again, it's the FDA and Health Canada approvals that will trigger capacity opening up. We don't have any concerns about getting capacity because a strong and compelling test is a very, very attractive thing to these contract manufacturers we're working with, who want fundamentally long production runs with high volumes. That's an extremely attractive thing for them, and we're not concerned about the ability to that we're going to have an incredibly well-performing test and not have sufficient manufacturing. The question is going to be, what does that ramp-up period look like, really? And that's something we'll speak about, and certainly we're working extremely hard on right now.
Thank you. The other question, has the test accuracy remained consistent throughout the latest field studies in the US?
We'll talk about those field studies when we have the reports back, and we very much look forward to that. But I will say that we're optimistic that we'll have similar performance of those tests. When you get into the hands, you take it out of the hands of our very capable scientists in our lab, and in the labs of places like MRI Global, a top-tier institute in Kansas City with whom we've worked, who've worked on the test and provided analysis and performance numbers on it. And into the field, there's always a degree of human error there in terms of the application of the nasopharyngeal swab that goes in and the transfer to the lateral flow test itself, the kit, the cassette. And this is frankly why in consultations with the FDA, ourselves, the FDA, and MRI Global all coalesced around the idea that, for this test, because it's a point of care test, really does need to be studied and validated in the field, in its operating context. So there may be some fall off, but look, we think that the test is extremely strong and expect the same numbers. If there's a little bit of fall off, that's the nature of a point of care.
Of course it is. I'm sure you hear this question all the time, so I'll ask it for our attendees. What is the likelihood of Sona being listed on the NASDAQ before September 1st? Or, more better put, what are your plans, if any, to list on NASDAQ?
Sure, the plans are just as indicated in our last release, that it is something we are exploring, and we'd advise the market looking forward. The NASDAQ is an attractive market. The US is the deepest pool of capital in the world, and there are an awful lot of specialist life sciences funds in the US that either require a NASDAQ or a US listing, or certainly you get on the radar by being there. And so it's something we're looking at seriously.
There's quite a few questions here very specific to your competition, but could you maybe point out who you consider your competition in the market right now, and where you see yourselves in your stage of development in comparison to them?
Look, the reality is we don't see a direct competitor. There are lots of competitors out there. There are lots of alternatives, effectively, to using and buying this test. But as far as competitors, direct competitors, it needs to be a rapid test. Within 15 minutes, you find out, that gives you early detection, which means an antigen test, effectively, or PCR if you can do it, but it's hard to have those two things within 15 minutes. And something that can be done by with a limited amount of training, and can be done anywhere. Whether it's in a remote field location, remote communities in the north, anywhere, and that doesn't currently exist. There are other antigen tests out there, but they rely on readers that from what we understand, are size of a toaster, and are specialized equipment, requiring technicians who are trained with that particular device. And by the way, those are costly devices as well. However, I'm sure they're all excellent tests, and antigen tests, period, are the way forward for getting us back to the course of normal. I will say that.
So you touched a bit on that, and to answer someone's question here, have you set a price point for these kits?
Not as yet. Price will be a function of their performance. Frankly, we need to see those numbers validated, and that's going to have a big bearing on buyers and the value that they ascribe to it, really. So we will see when numbers come in, and we're certainly in discussion with key interested parties on this matter, but we need to have final performance in.
Excellent. Do you have the assets to carry the testing and the FDA approval, basically, the funds to going forward up until you're into, a mass production?
So, as I mentioned in the presentation, we're very fortunate and very thankful to the government of Canada and to Antigen for the 4.1 million they gave us, and it had a specific purpose, which was accelerating the test. And they've, of course, had very strong governance around that, and parameters in terms of understanding how the funds were used. And of course, every focus we've had in this business for the last 6 months has been on this COVID test. So we're fortunate to have that funding, and we don't anticipate funding to be a constraint here. We're also hoping to have results from our field studies soon, and be able to forge forward with the process.
Excellent. How would a vaccine affect the sales of your product, of the test?
It's a good question. Somebody pointed out to me, I think it was Darren in the lab, the idea that a vaccine can really change things for the better, and we hope one comes out today, frankly. But in the case of HIV, in 1984, they said it would be 2 years before a vaccine, and
Yeah.
we're still waiting. So, I think we have to plan for the worst and hope for the best. And that means that, it may be some time for a vaccine. Nonetheless, if, as we hope, one comes out today, it's going to take an awful long time to roll out, both domestically and around the world. So I think the world is going to be in a place where it needs testing and tracing for at least the next 12 months. Absolutely.
Excellent. And one last question, a little bit more technical, if you don't mind. How long do the rods last, and is there any degeneration with each use?
That's an interesting question. And they're a one-use product. Each cassette test is used once. So, there wouldn't be any degeneration period in that time. We have in our instructions for use on the device a period of time for which the test, the lateral flow cassettes, would be valid. And so it would be guided by that, and folks would have a sense of that. But it is not a concern or a risk in any sense. And we have a great ability to create as much of the gold nanorod as required.
Great. There's quite a few questions, but I know we're hitting our time limit. I see Paul's joined us. Paul, would you like to ask David any questions before we wrap up?
David, first of all, fantastic story. And it's not just the COVID-19. One of your slides showed all the potential that you have in the future. I mean, I'm thinking of roadside testing for alcohol and cannabis, that they need like quicker results, like live, things of that nature. One question, the status of your patent pending, is that close to being done?
Well, it's pending now, so we're effectively protected. As to the precise timing on it, how those processes go. There's a protocol that must be followed, and country by country and whatnot, and it's well down that path now. So we're confident that we have many things to work on, and that fortunately isn't one that we have to worry too much about, because it's in good hands right now.
Cool. And one last question. I mean, your approval with the FDA, once you get it, how fast can you get it up and running and like into the market?
Now that's the $64,000 question right there. And that's, if and assuming we get the FDA approval, which we fully expect to get, it would be a quick turnaround in terms of first deliveries, because as I've indicated, we are right now in the process of tech transfer with a key manufacturing partner of ours with whom we've been working for some time to make sure that the way is paved, and manufacturing will be able to kick in short order from any FDA approval, and then start ramping up from there.
Are you on the fast track with the FDA? I mean, because they specifically targeted some people helping with COVID, that they'll fast track them. Like the U.S. government threw $1 billion at Moderna, trying to get a vaccine like quickly. Are you anywhere in that in that spot where they're fast tracking you, for example?
Well, I don't think medical devices have a specific channel like that, but I will say this: I think the FDA, obviously, a lot of great thinkers there, and folks who are very experienced in these matters, who have become much more educated, of course, over the last 6 months as to the specifics on COVID and whatnot, and so are very, very well informed. So we think that will benefit us, because there's been a growing reception over the last, as I say, particularly 3 months, to what an antigen test is and why it's so powerful. At the start of this pandemic, there were launch of all sorts of antibody tests, which are great, because in the absence of an antigen test or a PCR test on demand, it was something, and something was better than nothing. But then, of course, the thinking has evolved, as we've all seen, not only on the testing, but the testing protocols, as there's been a greater understanding as to how this is going to unfold. And it's not just the one wave, but it's going to be multiple waves that need to be managed properly going forwards.
Okay, and my last question, I'm assuming that your product is going to be high margin to you.
Yes. Yeah, we expect it will be. Again, still finalizing manufacturing arrangements. It'll be a function, obviously, of that when we know, as well as the performance of the test and the price that would be appropriate, both in developed world and underdeveloped worlds, and the protocols to be attached to each of those.
Perfect. Well, we'll wrap it up at that. Thank you so much to everyone who logged in today. It's a very high number. We're very, very pleased. David, you have a phenomenal story. Good luck with getting everything moving forward. And thank you for putting this together, because you are going to save a lot of lives with this.
Thank
Everyone
you, Paul.
be
Thank
safe.
you. I really appreciate it. Thank you, everyone. Okay.
Thank you.
Bye.